Today I want to present our ongoing work on modeling and validating the missing links in circadian regulation of dormant cells, especially quiescent cells. As we know, in your body, my body, we roughly have roughly 3 times 10 to the 13th of cells. So it's a lot of cells. And actually, anybody knows what the percentage of cells are in the cell cycle while we are talking? It's very small percentage. So we know a lot about the cell cycle, but actually only roughly less than 1% of the cells in our body are in the cell cycle. So the vast majority of the cells are not in the proliferation. Let me turn on the pointer. So here, proliferation, we know a lot, but only 1% of our body cells are here. So the vast majority of cells are in any of these states. We call them dormant. So dormant has different flavors, quiescence, differentiation, senescence, apoptosis. Well, this is going to be gone. But the major difference between the green one from an older review a long time ago and the principle still holds here is this is bi-way traffic. So cells can go into the quiescence, i.e. dormancy from the cell cycle, and they can return when there are proper growth signals. The other states are irreversible. They are dormant, at least for now, especially the differentiation. In this context, we mean the terminal differentiation or senescence. Cells go into this dormancy, but there's no way they can return, at least in normal physiological state, they will not return. So it's irreversible. So quiescence is very interesting for us to study because that is more like a sleeping-like state. They can return and they are responsible for a lot of cell types in our body, importantly fibroblasts, lymphocytes, and arguably most of the adult stem cells and the progenitor cells are in this quiescent, reversible dormancy state. And unfortunately, these dormant reversible quiescent cells play a big role in cancer development, especially in metastasis. So dormancy is something very important for stem cells, normal physiological state for tissue regeneration, repair, also has this pathological risk for us to tackle because they can go back to form the full tumor again. And over the years, we know also from the literature, also this is data from my own lab, we know the cells can go into dormancy, especially quiescence. Not only the quiescence is a reversible, but it's really a heterogeneous state. So in this particular example, our cells in cell culture, fibroblasts, so 2-day serum starved, you can do either serum starvation or contact inhibition, right? Either you remove growth factors or there's no space for them to grow. Either way, they will go into this reversible resting state. So you can put them in 2 days, 4 days, all the way to 2 weeks. But the longer the cells remain in quiescence, doesn't matter, you just put them there, no perturbation, or put the cells in a microfluidic device, give the constant flow with the refreshing of a nutrient, you'll see the same basic idea. Cells go deeper into quiescence. So what do I mean by deeper? In this example, say 2-day starved cells versus 2-week, 14 days starved cells. And if you give them 20% of serum, that's growth factors, to re-stimulate them after these many days, then the 2-day shallow quiescent cells, after 19 hours of serum re-stimulation, 80% of cells will re-enter the cell cycle, indicated by EDU, which is a thymidine homolog, can get incorporated in the DNA once cells get into the S phase. So that's our marker, or this is the approach we use to indicate how many cells are exiting quiescence. Again, 2 days very shallow, 80% cells will re-enter cell cycle after 19 hours of serum stimulation. But 2-week starved cells, now you give them 20%. After 19 hours, only less than 20% of cells will re-enter the cell cycle. So they are deeper. Question.
May I ask, what are the different color dots mean?
Different color. Oh, this just means different time. This is 19-hour, 22-hour. That's the time we harvest the cells for doing flow cytometry. You mean these dots? These are all the 19 hours for all these different days of starvation.
I see.
But similar idea, you see, the longer the cells were kept in quiescence, they are deeper in sleep. They take a longer time to wake up. Just like, human has shallow sleep, deep sleep. Cells have the same thing. The longer you sleep, the harder you wake up in the morning. I'm not sure that's true for people, but that's true for sure for our cells. But those cells are not irreversible, because if you wait long enough, like 41 hours, almost 2 days, they almost 100% of the cells can return. It just take much longer time. So put into a illustration is sort of like you think there's more than 2 wells. Those are continuous. You have shallower state, shallower sleep, and deep sleep, and shallower and deeper. So this is really continuum. We have run a modeling and experiments. We have demonstrate there's a gradual regulation in these quiescence depths. But long story short, the idea is the longer cells are kept in quiescence, this sleeping state, they are deeper into quiescence. Then it take longer to exit, and also you need a stronger stimulation. So they are deeper. Not only take longer, you need to push them harder. And this is just the same data, not same data, same cells done in parallel. Instead of stimulating them at different hours, we give them different serum concentration. Basically, same idea. The longer you starve the cells, they are in the cellular sleep, they need a stronger serum. For example, if 2 days starved cells, you less than 2% cells, less than 2% of serum after a day can push roughly 60% of cells into the cell cycle. But if 2-week starved cells, you need 10% to push more than half, again, 60% back into the cell cycle. So you need a stronger push and longer time. Basically, we call them are deeper in sleep. And over the years, so because my lab is really interested in how cells go into sleep and go into deeper sleep, what regulates the depths or heterogeneity of cellular sleep. So over several years, we have identified this RB retinoblastoma E2F gene circuit plays a major role in regulating the cellular sleep depth. In the first place, this simplified version of the RB-E2F cyclin CDK network, this performs as a, we call them a bistable switch, as you can see from the double wire. Most of you are familiar with the bistable system. So this is the bistable system. They have a threshold determined by this nonlinear regulation, but mainly from RB repression and the other upstream signals. And but through this positive feedback and with this strong repression in the first place, you can create, we can demonstrate, you can create a model, the bistable switch. Then we test in experiments indeed, biphasic hysteresis, memory-dependent behavior of E2F activation. So E2F off we'll use throughout the talk as the marker for quiescent state, i.e. cellular sleep, and E2F on is proliferation. But then the threshold, the red one, to turn on this bistable switch means the minimum serum concentration or duration you need to turn on this bistable switch from E2F off to E2F on, that's this red one, is directly correlate with the depth of quiescence. So we have shown them in the earlier papers. Basically, the higher threshold to turn on this beta stable switch indicate that cells are deeper in sleep. The shallower sleep corresponding to the lower activation threshold of this beta stable switch. So I won't get into the details. We already published those works in our earlier work. But that's a quick summary. So the RB12 cycling CDK network can perform like a bistable switch. Then importantly is the activation threshold of this bistable switch determine the depth of quiescence. When we run simulation, we can see the threshold has no like shallow and deep. It's really continuum. And we have started to identify several pathways crosstalk with this bistable switch and gradually regulate the activation threshold. So today I'll give you one example of there's many pathways regulate and interact with this bistable switch system. The reason we think this is a core regulation, not only because we studied it, is because this is the first bistable switch in the cell cycle entry, mammalian cell cycle entry from quiescence. So that's the first one. So we have shown on single cell level before this bistable module, cycling these CDK4/6 till here, there's a monotonic or monomodal response to growth factors. Then to E2F become bistable. So somewhere in this transition, they convert from a monostable system to a bistable system. So this is the first bistable system in the cell cycle entry from quiescence. And we think this is important to make sure normal cells has two very well identified and well separated states, which is quiescence and proliferation. This is important for cell fate determination for normal cells because we cannot have our genome amplified 50% and decide to come back. You have to make a decision then carry out faithfully. So the decision making is very important. But then how soon or how easy they can jump out of a dormancy into here, that depends on many other signals interacting with this core modulation. Then one example here is the circadian regulation. Oh, by the way, if you have any questions, feel free to stop me anytime. So over the past several decades, we have seen circadian regulation plays a lot of roles in modulating the stem cell regeneration, tissue repair, and also for, because the dormancy is important for cancer metastasis and linking to the cancer metastatic formation. So circadian clock is a lot busier than this, but in a very simplified view, because for all these oscillation, you need a negative feedback. There are several coupled negative feedback in circadian oscillation. The core part is the BML1 clock, but this is negative regulated by this loop. Cry per come back and activated by BML1 clock, but come back to repress. Same thing here. BML1 activate Rev ERB come back to repress. So those two are our main players today. I'll get more into this, but just remember there's a couple of negative feedback of circadian clock. So people have shown, this is a nice review, there's many coupling of circadian clock machinery to the cell cycle is well established. There's more than this simple cartoon. So we know circadian clock regulates the cell cycle, how quick and how cells go into the proliferation. But there's not any, or if at least for me, I didn't find many evidence the circadian regulation to the quiescent state, right? Quiescence is not, the cells are not in the cell cycle, so it's the other way, although they are coupled by this RB-E2F bistable switch. But we want to know, because as we said, if the cellular sleep is heterogeneous, then the shallower cells are easier to come out of a sleeping state, go into the cell cycle for tissue repair and regeneration. So we think not only the circadian clock may regulate the cell cycle machinery, likely the circadian clock may possibly to regulate the dormancy state through the RB12 bistable switch. And indeed, there is a, again, possibility. For example, this is the same simple machinery for the circadian clock. The main driver is BML1 clock, but they're regulated by two, at least, negative feedback. And there are literature to indicate that BML1 can repress MYC directly. And MYC is very important oncogene, and also MYC is an upstream regulator for both E2F activation. And it's necessary but not sufficient. But anyway, it's an important activator for E2F, and MYC is an important activator for cyclin CDK activity. But MYC is directly repressed by BML1, so keep that in mind. And then RevERB can repress CKI, for example, P21. So P21 is a very strong repressor for cyclin CDK in early G1 cyclin CDK, cyclin D CDK4/6, or G1S or S-phase cyclin CDK, which is cyclin E and CDK2. So P21 can be repressed by RevERB. So a few years ago, there's start to have available very very specific agonists, the activators, small chemical drugs can turn on CRY or RevERB. So CRY first. So CRY agonists, we use two. In this example, is something called KL001. So this can has a high affinity binding to CRY and activate CRY activity. Because CRY repress BML1, BML1 repress MYC. So we think, okay, if we activate CRY in quiescent cells, then MYC may be upregulated. So that's what we saw here in our fibroblast cells. Not very strong upregulation, but it's mild, but however, it's statistical significant. You can see these after before serum starvation, the cells are in quiescence for 2 days. If you give them CRY activator, then you see DMX was controlled. It's a dose-dependent increase of a MYC expression level. So given MYC is a upstream activator for E2F and cyclin D CDK, so we think likely, because you give them an earlier push, then likely these cells will be easier to get out of quiescence. That means cells will be pushed into a shallower quiescent state if you give them CRY activator. Are you with me? So CRY can turn on MYC, MYC can help E2F turn on the switch, so cells will be shallower, easier to get out of quiescence. Okay, that's one prediction. Another one is RevERB. So RevERB directly repress P21. P21 repress those important cyclin CDK complexes. And those repress a brake to the engine. Okay, P21 repress cyclin CDK, which is an inhibitor for RB. RB is a brake for E2F. And if RevERB repress P21, negative, 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 we have 4 negative, that's 1 positive. That means if you turn on RevERB, then eventually you turn on E2F easier. Again, cells will be shallower in quiescence. Okay, so that's our hypothesis. So again, before we do the experiments to test the quiescent steps, if we verify if that's true in our fibroblast system, indeed, when we give them dose-dependent increase of RevERB agonist SR9009, we see there's a dose-dependent repression of P21 here. You give them in quiescent cells, you give them increasing dose of RevERB, P21 is diminished. So you remove the brake, and cells can get easier out of quiescence. So in our system, we did verify both drugs work, and they modulate the corresponding regulators of this bistable switch correspondingly. So we have very strong confidence. Okay, now if we measure the depth of quiescence by either or both, they should drive cells into shallow quiescence. So we were very happy at that time. So let's do the experiments and see what we find. Actually, in both cases, we saw cells are going deeper in quiescence. That's correct, and this RIVRB. I just walk you through if you're not familiar with this type of data. So this is the flow cytometry. Each curve is a histogram of roughly 10,000 cells. So you can see a clear bimodal. So this is use the EDU, but if we use E2F reporter, same profile. So EDU again is the thermally homolog and ortholog can get incorporated in the genome. EDU negative means it's quiescence. There's no DNA replication. EDU positive means cells are now go back in the cell cycle, go into S phase at least. You have EDU incorporation. Then without either drug, DMS, or control, for example, at 2% of serum, wait for a day, you can see 35% of cells go into the cell cycle with 2%. But to achieve similar EDU positive percentage of the cells, 35%, you need more than 4%, give 15 micromolar of this cryoagonist, even higher, more than 8%, if the cells were treated with even higher cryoagonist. So the more you activate cry with, more MYC are expressed, but you need a higher serum strength. You need more serum, means growth factor concentration to push cells. Similar idea for RIVRB, right? With the same dose which I showed in an earlier slide, you see P21 is decreased. With removing the brick, however, you still need a stronger serum dose to push cells back into the cell cycle. Very anti-intuitive. Or if you look at the same dose, for example, here 4%, without the drug, you have 58 in this particular assay can go into the cell cycle, but with drug now, you have little less and even fewer cells with the same stimulation. Same thing. Either you need a stronger push to achieve similar level, or the same push will give you a smaller return of cells going into the cell cycle. Cells were deeper now into quiescence with more MYC, with less P21. Surprising. Okay, so but that's only one drug for each. Maybe there's some, who knows, maybe drug has some side effects or some toxicity. But no cells were dying with measured toxicity within this dose. Cells were happy, but maybe some unknown effect. So we tried different dose, different drug. Instead of KL001, we have KL002. Different chemistry. For same thing for activating cry, similar pattern. You need a stronger push. Cells are deeper in quiescence. Same thing here. SR9011, another agonist for VRB. You have less P21. You need a stronger serum. Again, anti-intuitive. Okay, maybe those are drugs. Maybe drug has something we don't understand. So let's do this genetic way. So let's just ecotypically express cry directly without drug. So this is a quiescent cells, a fibroblast, 2-day starvation. You have basic basal expression level of cry and RIVRB, but we can overexpress this by transfecting the cells with expression vectors. So this is Western. You can see now we have cry, more cry, and we have control. We just put in the same amount of 10 microgram of M-cherry as a control transfection. There's no cry here. Or RIVRB, we can overexpress. Same thing, M-cherry. Okay, so now we give them not through drug to activate. We just give them more cry or more RIVRB to see if cells are shallower or deeper. Again, cells are shallower because there's less EDU. You can percentage. You can get into the cell cycle with more cry or RIVRB expression. So through both the agonists and direct overexpression of either activators, we saw cells go deeper into quiescence. Again, that's very different from what we expected. So what's going on? We don't know. The only thing we understand is a missing link there. So we know the input is Cry, RIVRB are activated. We know the output cells are deeper into quiescence. We don't know what happens in the middle. Although we started with some hypothetical links, we think, but that should give cells into shallower quiescence, but now cells go into deeper. How can we find the target, potential target of a Cry RIV-ERB, which is missing in our understanding of the circadian regulation of quiescence depths? Well, we have a hypothesis that's something linked to one or multiple of these nodes, because those are the actual RBE2F gene network. There's close to 100 nodes. That's a very difficult thing to do on a bench, because if you don't have a target, there's no way you can identify. There's just become hopeless. You have to have a, for me at least, to find a potential convincing potential target. Then we design experiments, which is much, take a lot of effort to do. But we don't know which one potentially will be regulated by Cray RIV-ERB to drive into deeper quiescence because it's missing. Okay, then how can we do this? So we think maybe we can use the computer modeling to help us to find the possible, the most likely candidate for driving deeper quiescence. So our way of searching for this missing link is following. We simplified this real network again into our earlier 5-module network: RB repress E2F, then turn on cyclin E, repress RB, positive feedback. Then MIG is upstream and listen to growth factors, and this is an early step of activating E2F by phosphorylation of RB to remove the brake, cyclin D. Okay, so these are the modules now, not necessarily just cyclin D per se, but that's something linked to cyclin D activity. So we think, okay, since both Cray and RIV-ERB can do the same effect, which is deepening the quiescence. So for now, we combine them, thinking they may share something in common, may or may not, but we search for the common features, and they may somehow affect cyclin D CDK or MIC or any other of the 5 modules. So we have 5 potential targets. And there are 3 possibilities to each target, right? You can either, they may activate a potential target, they may inhibit the potential target, they may do nothing to the potential target. So for each link, you have 3 possibilities. And total, we have 3 times 3 to the power of 5, over 243 possible topologies without thinking about the parameter values, just 3 to the phase, which is over 200 potential topologies to link this Cray RIV-ERB activity to potential regulation to this RB2F switching behavior. So that means we need to generate over 200 ODE models. And the good thing is, in my earlier time, we have a core module through many years of work and bunch verification. We know this performed reasonably well as the core ODE module for RB2F bistable switch. But how can we make this to incorporate all these potential regulations from Cray and RIV-ERB? So we just add one term to any of these 5 modules. Basically, this is a Hill function with, well, this is the Maclaurin-Manton, because the Hill coefficient is 1 here. Then the M is the sign. So -1 means this is the inhibition, 0 means no activation, 1 means activation. So that's an M. For each term, I have three signs. Then W is the effective synthesis rate to match with the effective synthesis rate in the original model. And I is this half saturation rate constants. Basically, this just control for how sensitive are this term to Cray and RIV-ERB concentration, because we have different dose, right? Different way to activate CRAN and RevERB. So we just apply this to all these 5 terms, which corresponding to MYC, E2F, cyclin CDK, cyclin D, cyclin E, and RB modules. Okay, then by doing this combination, we can generate 243 ODEs total. Then we just run the simulation. We just assign low dose or high dose, just give them 0.1, 0.5 here to start with. Then what we did here is we have 200 something rows. Each row is a model topology ODE. But since we have parameters, parameter values for the core network, we don't have parameter values for this added link. So we just using the random parameters, thinking, okay, we don't know where, but we just scan 3 log of space based on our core module baselines, then just see what is the most likely topology or the way of regulation from RevERB to give us 2 features. The first feature is the cells will go deeper. That means in the base model, that's roughly 1%. 1% of serum can drive cells into the quiescence, from quiescence into the cell cycle. Now need to be stronger serum push, means deeper or higher activation threshold. So that's one criteria. Second criteria is this is the normal cells, not a cancer. It still need to maintain this bistable switch. You cannot disrupt the bistable switch by this potential regulation. So we scan for both features, 1 and 2. And then we rank these topologies based on the outcome. How many, if we run it many times, what's the success rate of a given topology will maintain both features, okay, at 2 different RevERB concentration. You can see there's no single model standing out, but there's clearly some winner subsets in both cases. If you zoom into these winners, you can see they have different ways to regulate those 5 modules. But basically, long story short, you can see, well, at least for them, we can see for us is the cyclin D CDK4/6 is the most likely target to responsible for RevERB driven deeper quiescence while maintaining the bistable behavior. And you can achieve similar things in modulating the other ones. For example, by 40% of cases, you can downregulate MYC in its top 10 modules, and they or either downregulate or upregulate or don't change. You can achieve this. These are, for example, MYC sometimes can be downregulated. In this case, MYC is upregulated. In this case, MYC is not changed. It's a little bit almost not random, but not clear. But this is clear. You need to downregulate cyclin CDK. So that's a model prediction. All right, what that means is this is the most likely missing link. So the RevERB from the modeling through all this random search is to most likely will target this module to drive deeper quiescence, meanwhile maintaining the bistable switch in our system. Okay, let's see if that's the case or not. But that's interesting to us because cyclin CDK not only is a missing link from both CRY and RevERB, and also this one of the 2 cyclin CDK complexes can regulate RB. Cyclin D can do this, cyclin E can do the same thing. But why only the early G1 phase cyclin, but not G1S phase cyclin? Why cyclin D, not cyclin E? Cyclin E really, most cases, cyclin E, you better don't change or change either up or down. Cyclin E is clearly in the modeling is not the winner, but cyclin D. Why that's the case? So retrospectively, I think we have some ideas why that's the case, but let's see if the model is true or not. Can the model prediction hold in our experiment? So this again is the fibroblast is red embryonic fibroblasts. So we just see give them KL001, the cryoagonist, increasing dose as before. Same here, RevERB increasing dose. We know cells are deeper in quiescence. Then let's see if they what they change. Indeed, this is the Western data, but here is easier to see in the quantification with statistics. So both cyclin D, there are 3 cyclin Ds isoforms in mammalian cells, but cyclin D2 is not expressed in ref cells. So only Western cyclin D1 and cyclin D3. In both cases, at from quiescence and 10 hours into or 15 hours, 14 hours into roughly that's the start of S phase, you see the cyclin D1 has a dose-dependent decrease in response to the cryoagonist. Same thing for cyclin D3 and CDK4, CDK6. All the cyclin CDK complexes, all the components are downregulated in expression by cryoagonist and also by RevERB agonists. But neither of cyclin E, there are 2 isoforms of cyclin E in the mammalian cells, E1, E2, not expressed, not changed by cryoagonist, not changed, at least not consistently by RevERB. The kinase, cyclin-dependent kinase of cyclin E, which is CDK2, not downregulated, Only the early G1 phase cyclin D, CDK4/6 are downregulated, but not cyclin E and CDK2. So this is just a summary of the data using, we take the both time points to take the heat map. We have done other Westerns, not only the cyclin CDK, the kinases, we did phosphatase like PP1, PP2A, same idea that they are not affected. The only one affected, EDU, is our dose-dependent decrease of cell cycle reentry. That's how deep they are. So the depth of the quiescence is directly correlated or clustered with cyclin CDK behavior, but not the other things. So again, confirming the model prediction, indeed, cryoRevERB directly target cyclin CDK in our experiments. So, and we just started to confirm this, although all our model, the base model at least, were constructed using the data from ref cells, but we want to see if this direct regulating of cyclin CDK can be potentially expanded to other cell types, because these are fibroblasts. Another major cell types in our body is, you all know, is epithelial cells. We cannot survey every single epithelial cells, but we have a convenient model in our lab too. It's RPE1. Some of you may we use the same model for epithelial cells. It's retinal pigment epithelial cells. So those cells have a good quiescent state as well. When your serum starvation or contact inhibition, you push cells into quiescence. So again, we treat the cells with, we haven't finished this one yet, but this is SR009. Very similarly, you'll see cyclin, maybe it's easier to show here. You'll see the EDU, again, it's a depth of quiescence, is linked for sure to PRBRB, which is a phospho-RB, and that's indicator of a cell cycle reentry, linked to the depth of quiescence. So increasing of SR009, again, that's cryo-ERB, cells go deeper. So both, they become diminished. But then they are linked to cyclin D1, D3 activity, CDK6 activity, but not cyclin E and CDK2. So it's a very similar targeting here in the same drug in another different cell type. Okay, so we are testing now into the cryoagonist, but so far everything holds very nicely. So I guess to summarize, basically what we found is the RevERB and the important circadian proteins. Not only they repress MYC, repress P21 as we confirmed, now they actually has a unknown inhibition of this important module, cyclin D CDK4/6 in the network. Although we don't know if necessarily this is direct inhibition or indirect, but we know the missing link, and now we identify those from both nodes here. The reason we think cyclin D CDK4/6 is important to regulating deeper quiescence rather its brother or sister, which is cyclin E. Okay, now there's a little bit hand waving. Okay, that's what we think. Although we have some modeling supporting data, but we don't have experimental data to demonstrate. The idea is this. So cyclin CDK is very important for this double negative, means positive feedback for this bistable module. If you break cyclin E CDK for CDK2, you have, remember these top 10 modules, you may perturb those to achieve deeper quiescence, but the parameter space is very narrow, because very easy you can break, if you perturb here to drive deeper quiescence, meanwhile, you may reduce this positive feedback strength, which may very likely disrupt the proper bistable control. So it's very hard to maintain a both required bistable switch. Meanwhile, drive cells deeper by perturbing this node. Cyclin D CDK4/6 is upstream of this feedback regulation. So this one is important for phosphorylating RB to remove its inhibition on E2F, but it's relatively independent of this feedback regulation. So you can safer to perturb this node or this module to drive cells into deeper quiescence without disrupting the bistable behavior of this E2F switching activation. So that's easier to decouple the task. So we think that's why they choose to target this one, but not that one. Although they eventually perform the same function to remove RB, but this is easier to be modulated. So, but if it's circadian, because we are doing cells in culture plate, there's no circadian regulation in cultured cells, although internally all the cells have all those proteins, so they are just unsynced. So, but in vivo though, then RevRB turn on in the late morning, roughly, and Cry and Purr turn on in the late afternoon, early evening. That means if we can predict, that means either in the morning or late afternoon, basically in the daytime, cyclin D CDK4/6 are repressed, cells are in deeper quiescence. But at late night or early morning, when both RevERB and Cry and Purr are low, neither is high in late night, and cells will be shallower into quiescence. So you may see a circadian oscillation of the quiescence depths, which may explain for why the stem cells have circadian, the regeneration power is different at different time of the day. So what we did is we sync our cells in culture plate. So we give them to our what's the drug name? I even forgot. So we call them DEX. So that's the hormone, the thing you can synchronize the cells. So then dexamethasone. So then cells are in quiescence. You have the dexamethasone, and they are also synchronized for circadian machinery. Okay, all this that now they are in the same phase in the culture plate. Now we give them, we just measure without perturbation, just measure naturally. Can you see a oscillation of EDO positive, means the depths? Indeed, when cells started, they're relatively deeper in EDO, but then 18 hours after release from the dexamethasone, they become shallower into quiescence. Then deeper again, you do see the oscillation behavior of quiescent steps in our in vitro system when you synchronize cells. So I think if we have an animal model to further expand this, the likely we will see the day and night oscillation of some stem cells go easier or harder for tissue repair and regeneration. This may potentially explain why people have seen, depending on the tissue, and some tissue, like some surgery is better done in the early morning because it's easier for tissue to repair and so on. And yeah, so basically there's a dimmer switch. Cells can go upper, shallower and deeper depending on the time of the day, how the circadian is regulating this bistable switch, changing the activation threshold. So in the end, just thank the people in the lab that doing this work. This work is both the modeling work started by Matt and followed by Xia, and the work in the fibroblasts were done by Sarah, and now the new work, and we confirming this, try to confirm those in epithelial cells are carrying over now by Chung. And thank you all. I thought I wouldn't take 50 minutes, but almost 50 minutes. Thank you very much.
All right, let's thank Guang for this very, very interesting and wonderful talk. So if you have any questions, you can put in the chat. And maybe I can start with some questions. So I'm wondering if you have, for example, try to match the level of variations between the modeling and the experimentation, because apparently you talked about heterogeneity, right? So there are this percentage. So have you like matched those variations quantitatively between the modeling and the experiments?
Not directly in this particular work, but early on we have done those exercises. We have this not ODE but SDE, and we do see there's a very good correlation between the SDE prediction to the percentage of EDU positive. So that is nicely matched.
Cool. So please, I mean, you can also like raise your hand or directly unmute yourself if you have a question. Oh yeah, I see a question from Garima. Garima, would you like to unmute yourself and ask? Okay, I can also read out the question. It was a good talk. So what happens to the other important players of circadian biology, like clock and BMAL, upon the introduction of agonist for the negative regulators of the circadian biology? And there's a second question, how will this affect the other important genes which are under the regulation of clock? So maybe first question first, yeah.
Yeah, right. So definitely. So when we didn't measure directly in our experiments, but when where's my mouse? Here. Oops, sorry. I'll go back 1 second. So we think, okay, we didn't verify this. So when you turn on RevERB, BMAL1 will be inhibited. Same thing when you turn on CRY, the BMAL1 clock activity will be repressed. And those will actually, for example, that's why MYC is repressed. Without the drug, and with drug is de-repressed now, have a moderate but significant increase of MYC activity. So we think that's through this, but we didn't verify them directly. We just think because this is well established, that should be the way to work. And also we are focusing more downstream. We didn't verify the links within the circadian clock machinery. But that's important if you really want to dive into what's going on with the CRY or RevERB. Agonists are changing within the circadian machinery. So how will this affect the other important genes which are under the regulation of the clock? So yes, the clock regulate a lot of genes. Many are important for our body physiology. And also I showed the earlier link, the cell cycle has DNA repair machinery, cell cycle progression are regulated by the circadian clock. So again, we predict if we perturb this network by either agonists, these machineries will be affected. But we didn't test because we are mainly focusing on the entry from sleep to the start of S phase. We didn't go all the way to in the cell cycle and DNA repair, but we would think they will be affected by those perturbations as well.
So I have another question so related to this diagram. Basically, this seems to show that for instance, the reverb, is both inhibiting and indirectly promoting psyche, right?
Mhm.
So I mean, what do you think is the kind of signal response that can be generated by that kind of double regulation?
I see. So you're referring to this directly repress cyclin CDK, but through
Yeah.
the P21 can come back. Yes. So we did see both. P21 is downregulated, but this is also downregulated. In the end, is a balance. So you have activators and repressors depending on the efficacy of each one to change the quiescence steps through the activation threshold. And so in short, in the end, is this probably because this is more direct, maybe, but not necessarily. But this has a stronger net effect.
So do you think there could be some like temporal effect, like because the
Ah,
pathway between reverb and cycling D, you don't know if it's direct or indirect, but
right, right.
so in terms of the time scale, so have you tested it if there could be some kind of temporal
Yeah,
effect?
yeah, good question. Yes. So, because indeed, this basically is incoherent feed forward, right? So that there's maybe temporal, maybe you can see something repressed and deep repressed,
Mhm.
and very likely we didn't. So, but if we follow ourselves longer enough, right? So probably we will see some of these temporal regulations as well. Now we
Mm.
only see, can you enter or not?
Mm.
Yes.
Zanya, you have a question?
Yeah, I had two actually.
Okay.
I can do them one at a time. One is not that scientific, more just out of curiosity.
Right.
How long computationally did it take to do the, 243 models times 10,000 parameters? Just
Oh,
um,
right. That one, because we have HPC, the high performance computing. So we
okay.
do, yeah, no, it's a couple days or
Okay.
not
Cool.
even quicker if we do them in more parallel manner. Yeah.
Wow. Okay. Um, that's
It's much quicker than doing experiments for sure.
yeah, well, yeah, that yeah, that makes sense. Um, And then my more, scientific question is I'm intrigued by the bistability and the hysteresis in the RBE2F switch.
Right.
And I'm wondering if it's known whether there's any natural biological role that plays, or if that's not known yet. Um,
So you mean the natural biological role of this vestibular switch in,
Yeah, I mean,
right.
if there's any biological situation where the hysteresis might occur, outside of an experimental situation. Um,
The that's what clarifies you. The hysteresis occur in this RB12 gene circuit, but uh, outside of the cell cycle regulation, or I just want to get,
no, well, I guess I'm thinking that because you can observe the hysteresis in the experiments,
yes.
but I'm wondering if it ever would have a natural biological role, in situ or outside of the experimental conditions.
Okay. So yeah, I think I got what you meant. So I think naturally this hysteresis is very important for this cell cycle reentry from quiescence. And it's very important decision to make. And that's where all cancer cells become cancer. Is they have one way or the other something wrong in this bistable regulation. So it becomes little too easy to go to jump out of quiescence and go into cell cycle, and probably harder to return if you can draw. It's like this. So, and because this thresholding or the hysteresis, and this may even, again, I haven't done this experiment, but may just from literature, it may explains for this potential even this not full, because the hysteresis is you drive cells into the cell cycle in the S phase. And even now the environmental signals are gone, right? The factors, they will at least truthfully to finish the current cell cycle. That's very important for normal cells. But now, if you only have genome amplified, not fully done yet, but then you rush it over, or you don't have hysteresis when environmental signal fluctuate, the cell cycle be cut short. This will cause a lot of problems.
Yeah. Okay. Thank you. Yeah, that makes a lot of sense. Thanks.